So in this section, we'll be looking at the structure of the genetic material. Before we were talking about genes, how they're transmitted, and heredity. This is the structure of what does a gene look like, and how does it affect phenotype. Experiments have shown that DNA is the genetic material. Hershey and Chase in the 1950s showed that certain phages, and phages are just a very simple bacterial virus, were able to reprogram host cells and produce more phages by injecting their DNA. And the following is the experiment that they did to show that. Now, they knew that phages were composed of proteins and DNA. That's the only thing in them. They're very simple organisms. And they were aware that the phage attaches itself to the bacterial cell. And they were trying to figure out whether it's a protein that caused it to be able to take over the cell machinery or whether it was the DNA. So they did the following experiment. They made in the batch 1, they made the protein that's shown in yellow radioactive. So the protein coat on the outside is radioactive. When they centrifuged them, the empty protein shells fell off the outside of the bacteria, and we got a pellet here that had no radioactivity. And the radioactivity was found in the liquid. When they made the DNA radioactive, however, they found the DNA was associated with the pellet. And the DNA was radioactive, of course. And that is because the DNA was inside of the bacterium. So this is the life cycle of a phage. This is a phage. Looks a little like a Martian lander. It attaches itself to the outside of the bacterial cell. It injects its DNA into the cell. The protein shell remains on the outside and eventually moves away. The phage DNA directs the host DNA to make more phage DNA proteins. So the cell stops making proteins that might be useful to itself and instead is hijacked and makes only phage DNA. Once it makes the phage DNA and the phage proteins, the cell is lysed and new phages are released, and so it can go on to infect another cell. So only the DNA is injected into the host and the protein stays outside. Now we know now that DNA is a double-stranded helix and it's composed of nucleotides. It contains a deoxyribose, which is a ribose sugar with an oxygen missing. Deoxy just means without oxygen. And a sugar phosphate backbone. And then it also has these funny looking base pairs. So Watson and Crick are the people who worked out the three-dimensional structure of DNA. And the structure is 2 polynucleotide strands wrapped around each other in a double helix. And a double helix could be described as a twisted rope ladder. So here we have the sugar and phosphate backbones, and these are the base pairs, one coming from each side, and then they're twisted together. They won the Nobel Prize along with Wilkins in 1962. Rosalind Franklin probably would have received credit as well. She died of ovarian cancer in her 30s, probably because of all the radiation exposure that she received at work. And she was responsible for this picture here that looks kind of blurry and probably doesn't tell you or me a whole lot. But when Watson and Crick saw it, they immediately saw that the structure of DNA was going to be a double helix. And then they had to put it all together to figure out why. When they first started trying to figure out this structure, they thought that it was going to be 3 strands and that the bases, because they had the hydrogen bonding, that they would be on the outside. So that they were trying all sorts of different things to try and get these nucleotides to fit together. And they did some pretty cool math to show their structure worked. The Watson and Crick model gave new meaning to the words gene and chromosome. The genetic information on chromosomes is encoded in the nucleotide sequence. Now, the strands are held together by hydrogen bonding. We're about to see a picture. And each base pair has a complementary partner. So adenine always goes with thymine, and guanine always goes with cytosine. And I usually remember these because A goes with T, and G looks a little like a C. The G and the C go together because they both got that curved shape. I don't know if that helps or not. That's just what I use. Okay, so here are 3 different models of DNA. The first is the ribbon model, and you can see the sugar backbone, sugar phosphate backbone, and the bases are coming out here. Here's a T, and it's with an A, and here's an A, and it's with a T. Then we have the partial chemical structure. Here, the sugars are drawn out. That's a sugar, if you'll recall from earlier, and that's phosphate. So we have sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And in the middle, we have our base pairs, and you'll see that C is going with G, and that A is going with T. And the last one, I think, is probably the least useful, but it's a computer model. It's more of a space-filling model, what it would actually look like.